Hello once again. This is video 5, and C3 is the last of the material for test 3. And hopefully we'll get it all done on this video. So we are going to begin with cellular respiration. How cells are going to be converting the glucose, the chemical bond energy, into ATP. So let's just review this a little bit. Probably recognize this picture. This is a chloroplast, and the light reactions, photophosphorylation of the light reactions, those take place in the thylakoids. They're going to be producing ATP and NADPH. Let's leave the thylakoid, go to the stroma, still in the chloroplast, and the ATP and NADPH are going to be used to reduce carbon dioxide into glucose. Glucose, and this is where we're starting for this video, glucose now leaves, goes to the cytoplasm. Glycolysis, splitting of glucose, takes place in the cytoplasm. There are two pyruvates that would form. And those pyruvates are going to enter into the matrix of the mitochondria. So glucose, which is 6 carbons, so glucose, oops, little G there, glucose gets converted into energy. There you go. Right? Energy. So glucose, chemical energy, chemical bond energy, is going to be converted into what kind of energy? Phosphate bond energy, right? So that is going to be converted into ATP, phosphate bond energy. So glucose, C6, H12, O6, is also going to be using oxygen. And we're going to be seeing that oxygen is actually going to be only used at the very last step of cellular respiration. But it is extremely important, as we know. So we know we need some oxygen. Alright, another arrow here. And notice we have a big molecule, glucose, and we're going to break it down. So if we break it down, that's oxidation. So in this case, glucose is going to get oxidized. And it's going to get oxidized back into what we started with in the Calvin cycle. 6 carbon dioxides and water. Okay? So this whole process, we're going to add a lot of details to this. And probably 6 waters here. Let's put 6's in here because they're all 6's. So this is a short summary of cellular respiration. Now, when you think of the word respiration, you probably think of breathing. This is actually going to be related to breathing. Notice we have oxygen. These are the reactants, so we need oxygen. Oxygen that we're going to be needing is coming from the atmosphere. And we're going to be exhaling carbon dioxide, which is one of the products of cellular respiration, and we're going to be exhaling that. We're going to be releasing that back into the atmosphere. So let's look at this word metabolism. Basically, those are all the reactions that are taking place. And we're going to review some words that we used before this: metabolism, Like catastrophe, remember that? Anabolism. Like catabolism, breaking, anabolism, building. So metabolism are all the catabolic and anabolic reactions of an organism, so making, breaking bonds, basically is what it's saying. So catabolism, big molecule, into smaller ones. Breaking bonds, so releasing ATP. That ATP is going to be used in anabolism, small molecules being built into bigger molecules. So let's relate that to reduction and oxidation. Reduction, which one is this? Which one is going to be reduction? Remember reduction, you're going to receive it, so reduction would be this. Anabolism, building. Oxidation, losing. So big molecules becoming smaller molecules. And ATP is going to be made during catabolism and actually going to be used during anabolism. So making is like charging a battery, and then you got to charge the battery up again, and then you use it, charge it up again. So ATP is kind of like a battery. And if you remember, ATP is the only molecule that we can use for energy. It's like the currency of our body, ATP. So once again, ATP is made during catabolism and used during anabolism. So making energy during catabolism is like charging the battery. ATP molecule like a rechargeable battery. When it's fully charged, it's ATP. It's run down, it's ADP. So again, anabolism, this is our synthesis stage. Simple substances are synthesized into complex molecules to store them energy. Energy is required. You're going to be making bonds, you're going to be building up. Complex molecules form from simpler ones. Catabolism, catastrophe, breakdown of complex molecules into simpler ones, often resulting in a release of energy because you're breaking chemical bonds. So releasing energy. Living tissues change into energy, waste products of a simpler chemical composition. So let's review ATP. Remember A, adenosine, tries 3. Remember these are wavy bonds, high energy bonds. High energy because the phosphates are negative, they do not like to be next to each other. It takes a lot of energy to hold them together. So whenever you break one of those bonds, energy is released. A little explosion thing means energy. It's just the right amount that your cells need. Now we have ADP. And one more energy bond. We can actually break that off and make AMP. Whenever you break one of those bonds off, you release just the right amount of energy. So catabolism. Notice this one does not have a high energy bond. It's not a wavy line. So that one is not going to be broken off. But now we can remake ATP. So catabolism was the first part when you're breaking the chemical bonds, you're using the battery, energy is being discharged. And now we're going to recharge the battery, and that is going to be through anabolism. So here, putting energy in the phosphate, right? ADP, put energy in. Put another phosphate to make ATP. Requires energy to put them together. So now we have ATP. So again, ATP is extremely important. It is the only molecule our body uses for energy. So this whole cell respiration is about making the ATP. So making ATP, like getting some money, using it, using the money, and you're breaking it down, and you gotta make some more money. And let's review oxidation and reduction reactions, and here's another way to look at it. Oil, rig. Oxidation is losing. Rig, reduction is gaining. However, it helps you to think about it. But remember, in biology, we're talking about three things that are going to be either lost or gained: electrons, hydrogen, and energy. Respiration is going to be oxidative breakdown. Glucose is the #1 molecule that's going to be used, but we will see that we can actually use fats and proteins. Goes in each cell, and that's how we're going to be making the ATP. So photosynthesis, so which we began for this test. Carbon dioxide and water. Water was oxidized. Carbon dioxide was reduced. So carbon dioxide was reduced in the Kelvin cycle, and it became the glucose in the Kelvin cycle. Water was oxidized, gave off electrons which were used, gave off the oxygen that went into the atmosphere. Alright, so electrons, oxygen, hydrogens, which could have several important jobs, maybe involved in chemiosmosis and the hydrogen pump used to make ATP. Respiration. Now we start with that big molecule, and now we're going to use oxygen if it's aerobic. Notice 6 carbons here becomes 6 carbons of the carbon dioxide. Up here in photosynthesis, 6 carbon dioxides ended up on the glucose. So respiration, glucose is going to be oxidized and become carbon dioxide. Oxygen is going to get reduced and become water. Here it is, oxidized, reduction. And you may have noticed that photosynthesis and cellular respiration are actually opposite. So I put an arrow here. Let's put that one up. There, arrow. Oh, didn't work. So on the top arrow, look, I just made it opposite. I'm going to change this a little bit here. And we're going to say this is solar energy. Some of the solar energy is going to be converted into chemical bond energy in the form of glucose. And carbon dioxide gets reduced into the glucose. Now notice, we're starting out with this. Notice they're just the opposite pretty much. Glucose. So this is the products. Now these products are the reactants. Plus oxygen. Now the glucose gets oxidized, right? Comes carbon dioxide. Goes for waters. And depending on the organism, 36 in a eukaryote, 38 in a prokaryote, like bacteria. So basically they're just opposite reactions. So if you know photosynthesis, you're going to know respiration is just going to be opposite. So solar energy gets converted into chemical bond energy in the form of glucose. Chemical bond energy in the form of glucose becomes phosphate bond energy in the form of ATP. So the whole process was to convert solar energy into ATP. Almost all the reactions in our body require ATP. Respiration is actually quite complex. We're going to keep it simple. It's a series of step-by-step reactions. Every single step has a different enzyme. Don't worry, we're not going to learn any of the enzymes. And it has to happen in a certain sequence. We have 2 electron carriers that are going to be involved in cellular respiration. The first one is NAD+. Remember, photosynthesis had NADP+, and now it's NAD. So P, photosynthesis starts with a P. Okay, maybe that will help. So NAD+, those are the ones that are going to be carrying electrons, hydrogens, and energy. The metabolite is oxidized. NAD+ accepts 2 electrons, 1 hydrogen, and becomes NADH. Just like the taxi, just like NADPH. The electrons it receives are usually going to be carried to the electron transport system. The second one, which we have not seen before, is FAD. So this is another electron carrier. And this is going to receive 2 electrons, 2 hydrogens, and it's going to become FADH2. These electron carriers are extremely, extremely important. Right? When they give their electrons, hydrogens, and energy to the electron transport system, that is going to be used to make most of the ATP. So these electron carriers are really, really important. Before we get started, let's review a mitochondria and what it looks like. Alright, so that is an image of a mitochondria. Remember, mitochondrion, singular. And it's going to be important to know that it has 2 membranes. Then intermembrane space is going to be important. The cristae right here, it's folding right here. And the space outside it, the fluid space, that is called the matrix. So look what I have down here. We haven't gone through all these steps yet, but you can come back and review this. Glycolysis, so glucose, in the case of the plant, glucose left the chloroplast onto the cytoplasm, and in the cytoplasm right here, glycol, sugar, lysis to split, glucose, which has 6 carbons, is going to be split into 2. And in that process, it is going to be making 2 NADHs and a net gain of 2 ATPs. When the glucose gets split into 2, it is converted into 2 molecules of pyruvate. Pyruvate have 3 carbons each. And then that is going to go into the mitochondria. These next 2 steps are going to be in the matrix of the mitochondria. We have a transition step where acetyl coenzyme A, acetyl-CoA, that's a C, is going to be giving off a carbon. And that is now going to go into the Krebs cycle. Right? And these are going to be making electron carriers. So electron carriers are made in glycolysis, transition step, and the Krebs cycle. Krebs cycle makes a lot, most of them. And all of those electron carriers go to the electron transport system, also called electron transport chain, which is in the cristae. And here we're going to see chemiosmosis is going to take place again. That's with the ATP synthase, the hydrogen pump, and that is where most of the ATP is going to be made. Here's another way to look at it, a little simpler version of it. So this is the mitochondria. Let's start with glycolysis. Glucose, let's say it's us, could be some food that we ate. The glucose, which has 6 carbons, gets split into 2. And it gets split into 2 pyruvates. The pyruvates are going to go into, so those are out here, let's say this is the cytoplasm. Then they're going to go into the matrix, matrix. Right here. Right here, we see that teal color, greenish color. We go into the matrix. The matrix is where the transition step takes place and the Krebs cycle. The Krebs cycle is also called the citric acid cycle or the citrate cycle. So, most two common names are Krebs cycle, citric acid cycle. And they're all making their electron carriers. As soon as the electron carriers are made, they're going to go to the cristae. This is that purple. So they're going to go to the cristae. And the cristae is where oxidative phosphorylation is going to take place. That means it uses oxidase and reduction. It's going to be using electrons. So glycolysis makes electron carriers. They get sent over here. Transition step makes electron carriers. They go to here. Krebs cycle makes electron carriers. Send them over here. Folding cristae. And they're all used to make ATP. Remember prokaryotes. They do not have a mitochondria. So they're going to have a slightly different number of ATPs that are going to be made. Even if you can't see this picture that well, I just want you to focus on this part here with the red circle. So the amount of ATPs, right here, 38 ATPs for prokaryotes, 36 for eukaryotes. And that is because the prokaryotes do not have a mitochondria. Look at this 4 to 6. In eukaryotes, they're also making 6 ATPs, but they're using 2 of them. Kind of like paying a toll to be able to use the mitochondria. Prokaryotes do not have to pay that toll, so they're going to make about 2 more ATPs than the eukaryotes. So we just have that in words. It's kind of like the eukaryotes have to pay a toll of 2 ATPs to actually get into the mitochondria. Prokaryotes do not have to go into the mitochondria because they don't even have a mitochondria, so they don't have to pay the mitochondria access toll. So each of their NADH produces 3 ATPs, and none of them are used as a toll, so they're going to make 38 ATPs versus 36. The 4 stages of aerobic respiration we're going to go through. Aerobic means the kind that uses oxygen. There's glycolysis, splitting of glucose. So 6 carbon glucose gets split into 2 3 carbon pyruvates. Alright, those pyruvates go into the transition step, so now they're going to go into the matrix of the mitochondria. They're going to form acetyl coenzyme A or acetyl CoA. That molecule now goes into, still in the matrix, the Krebs cycle, also known as the citric acid cycle. All 3 of these first steps are going to be making electron carriers, and all the electron carriers go to the electron transport system, where chemiosmosis is involved in making the ATPs. So let's look at this again. Look at the ATPs. Glycolysis makes 2 ATPs. Transition step makes you know it does not make any ATPs. Krebs cycle makes 2 ATPs. And notice, as I said, the electron transport system makes most of the ATPs, and here there are 32 ATPs. So let's look at this again. 2 ATPs over here. Glycolysis, those are made through substrate phosphorylation. Transition step, no ATPs. Krebs cycle also makes 2 ATPs using substrate phosphorylation. So substrate phosphorylation, they're not using electrons. Taking a phosphate off of a molecule, off of a substrate, and putting that onto ADP. And 32 ATPs are going to be made through oxidative phosphorylation in the electron transport system, almost exactly like in photosynthesis. So glycolysis, cytoplasm, cytosol, you can use either word. And all the rest of it takes place in the mitochondria. These two right here, transition step and the citric acid cycle, take place in the matrix of the mitochondria. The electron transport system is in the cristae of the mitochondria. So here's another way to look at it. Catabolic pathways. Aerobic. Think about air, right? Oxygen, air, aerobic. This actually needs oxygen. And this is going to give you the most ATP. And if you look at the picture, all the arrows going straight down, that's aerobic. Anaerobic, without oxygen. Oxygen can be present. Some organisms just do it this way. But we can do either way. And our cells will have oxygen sensors telling how much oxygen is there. So if there's not that much, then it will go to, in this case, lactic acid formation. So anaerobic, we can have alcohol fermentation, right here, or lactic acid fermentation. So these are anaerobic. And we'll get into more details later on. So let's look over here, glucose. Notice it has 6 carbons. It's split into 2 pyruvate, so actually many steps in between here. And each step has its own enzyme. And it gives off 2 ATPs. It's also giving off the electron carriers, but this picture is just dealing with the ATPs. Now, this is anaerobic, so even in our aerobic cellular respiration, the first step is anaerobic. Alright, let's say there is enough oxygen present. So this pyruvate, alright, you can tell, there are little sensors in there to tell if there's enough oxygen. It will leave the cytoplasm, go into the matrix of the mitochondria. So everything below this line is in the mitochondria. Notice that has 3 carbons. This is acetyl group. It's going to join in form over here, acetyl coenzyme A, or acetyl CoA. And it's going to give off 2 carbons in the carbon dioxide. So each pyruvate is going to be losing a carbon. So it's going back into the carbon dioxide. Now it's getting oxidized. And that's some of the carbon dioxide that we're going to breathe out. So that's a transition step. That transition step goes into the Krebs cycle. And the Krebs cycle is going to go around twice. Just like the Calvin cycle. And in that process of going around twice, it's going to make 2 ATPs. And look over here, 4 carbon dioxides. So now the glucose is completely broken down. All the carbons, the 6 carbons that were in glucose, are now back in the carbon or carbon dioxide. Each of these steps, glycolysis, transition step, Krebs cycle, are all making electron carriers, and they go to the electron transport system where most of the ATP is made. We're only going to talk about eukaryotes in this class. 32 ATPs. So 2 ATPs up here through substrate phosphorylation, 2 down here in the Krebs cycle through substrate phosphorylation, 32 down here through oxidative phosphorylation using chemiosmosis, electrons, electron carriers. A total of 36 ATP. That number actually will depend on the health of the cell, other things, but 36 ATP are possible. So right here, let's look at this very last step. What gas are we breathing in? We are breathing in oxygen. And oxygen is used right here at the very last step. Oxygen is the final electron acceptor. Oxygen is going to get reduced. It's going to gain electrons, hydrogens, and energy. And guess what happens? The oxygen that we breathe in becomes water. Look at that. So the oxygen that we breathe in becomes water. Remember in photosynthesis, the water was oxidized, right, and it became oxygen. Now the oxygen is getting reduced and become water. So first we're going to deal with that, and then we'll go to this aerobic part. So here's another picture. Glucose, glycolysis, orange pyruvates. There's 2 ATPs here that are going to actually be used. 2 of them are going to be used. 2 NADHs, electron carriers, 4 ATPs. Transition step, now we're in the mitochondria. All this pink stuff here is the mitochondria. 2 NADHs, 6. Krebs cycle goes around twice. These are the results of going around twice. 2, 18, 4. Alright, let's look at these electron carriers. We have 2 NADHs here, 2 in the transition step, 6 NADHs. So these first 3 are NADHs. Not NADPH, but NADH. And this is the very first time we're going to see FADH2. This is actually the only time we're going to see it in this class. There's a little notes to it. So this is substrate phosphorylation, substrate phosphorylation. This down here. Okay, so look right here. The reason why these are here, because these are not, I should point this out. Just make sure you understand this. These two here, notice, these two, this one right here, and this one right here, does not have any electron carriers here. See that? No electron carriers. So this is making 2 ATPs through substrate phosphorylation. This is also making 2 ATPs through substrate phosphorylation. But they're also making electron carriers. These electron carriers, these are the ones they're going to use in oxidative phosphorylation right here, this. So all these other ones that have these electron carriers, these are going to be made through oxidative phosphorylation. So you're going to have a total of 2 plus 2 is 4 ATPs through substrate phosphorylation. And the transition step doesn't make any. You get a total of 36. Glycolysis, glucose split into 2 pyruvates, makes 2 ATPs through substrate phosphorylation. Also makes 2 electron carriers. Makes 6, but it's going to be using 2 of them. Makes 2 electron carriers in this. You know what that's called? That's the transition step. This is all now in the matrix. 2 NADHs, 2 electron carriers. Goes to the electron transfer system. Makes 6 ATPs. Citric acid cycle, also known as the Krebs cycle, makes 2 ATPs through substrate phosphorylation. It makes a lot of electron carriers. 6 NADHs, 2 FADH2s, 18 and 4. Now, let me point out something that might be helpful to you. Notice 2 and 6. So for every NADH, it's going to be making 3 ATPs. So if it's 2 NADHs, 2 times 3 is 6. Notice this is making 6 NADHs. 6 times 3 is 18. Notice here, this is different. 2 FADH2s only makes 4. So for every FADH2, we're only making 2. So FADH is going to make 1 less ATP than NADH, and I'll show you later how that happens. If we're actually in class, I would give you a diagram that looked like this, and we'd fill it out. And also on your test, you would fill it out, and your cumulative final, you would fill it out. So let's go through it. So, good way to study, be able to fill out this chart. So this is a nice little summary of what you need to know. This is aerobic, but as I said, remember the very first step, glycolysis is anaerobic. So the first step of aerobic respiration is an anaerobic step. It does not require oxygen. It takes place in the cytosol or the cytoplasm. Here, glucose, which has 6 carbons, is going to be split into 2 pyruvates, each having 3 carbons. So glycolysis, splitting of glucose. In that process, it makes 2 ATPs, that is through substrate phosphorylation. We actually talked about substrate phosphorylation in A3, but I will be reviewing that for you. And look at this, a number of steps. It could be 8 steps, 7 steps, 10 steps, and every single step would have its own enzyme. It's also making 2 electron carriers, and these arrows are showing you as soon as it's made, it goes over to the cristae of the mitochondria, where the electron is taking those electrons to the electron transport system for chemiosmosis to take place. So it's going to have a hydrogen pump, just like chemiosmosis in photosynthesis. 2 pyruvates now are going to go into the matrix of the mitochondria. So these 2 tealish bluish colors are the matrix of the mitochondria. Transition step. No ATPs are made. It does make 2 NADHs, which go to the electron transport system. 2 times 3, 6 ATPs. So here 2 NADHs, only 4 because remember it has to pay that mitochondria toll. So it makes 6, but it uses 2. There's our transition step, there's our transition molecule, acetyl coenzyme A or acetyl CoA. It only has 2 carbons each. So 2 of them each having 2 carbons. Why does it have only 2? Because look, it gets oxidized, it gives off 2 carbon dioxides. So that transition molecule goes into the Krebs cycle, goes around twice. As a result of going around twice, it makes 2 ATPs through substrate phosphorylation, and it completely now breaks down the glucose. 4 carbon dioxides are given off. So every carbon of the glucose is going to get released as a carbon of the carbon dioxide. Alright? Let's look at these electron carriers. 6 NADHs take their electrons, hydrogen, and energy to the electron transport system. So 6 times 3 is 18. 18 ATPs. 2 FADH2s take their electrons, hydrogen, and energy to the electron transport system. 2 times 2 is 4. Makes 4 ATPs. So add all these up. 32 ATPs made through oxidative phosphorylation. That's in the cristae. Substrative phosphorylation makes 2 here in glycolysis, and 2 here in the Krebs cycle. So substrate phosphorylation, looking down here, glycolysis makes 2, Krebs cycle makes 2. Over here, oxidative phosphorylation, electron transport system makes 32. So from 1 glucose, we get 36 ATPs. Since most of the ATPs are made in the mitochondria and the cristae, The mitochondria are called the powerhouses of the cell. So the only three places where ATP is made: glycolysis, Krebs cycle, electron transport system. Glycolysis and Krebs cycle make it through substrate phosphorylation. Electron transport system, chemiosmosis, makes it through oxidative phosphorylation. Alright, now we're going to go into each of these steps in a little more detail. Alright, this is a picture of glycolysis. I'm sorry I had to make it really small so that you could see the whole thing. But that's okay because you don't really need to know everything in here. Actually, hardly anything. So you're going to start out with glucose.